Please welcome Nikos Karaiskos, Berlin Institute for Medical Systems Biology, Max Delbruck Center for Molecular Medicine in the Helmholtz Association. Solving spatial transcriptomics puzzles.
Right, so hi, and thanks a lot for this wonderful opportunity. So I would like to start first with ordinary puzzles. And I would like that you ask yourselves a question, how do we solve a puzzle, like an ordinary puzzle? So imagine that I give you a puzzle, but I don't tell you what image you have to create. If you have a few pieces, then it's pretty straightforward to do it, and you can just reconstruct it. What if you have a bigger puzzle? What process do you use to actually create the picture? So you will try to take pieces which have similar colors and similar edges and assemble them together, right? If you have no colors in the picture, it will take you much more time, and then you are stuck with just matching the edges. Now think of a different problem. Let's say that you have a puzzle, and every color represents an image. So in the end, you don't have one unified image to create, but different images. And you have to do this for 100,000 pieces and 15,000 colors. So that's a quite daunting task, but this is exactly the problem we are trying to solve here. Because you see, single-cell technologies are really great, but the problem is that you have to dissociate this issue in order to obtain single-cell transcriptomes. And therefore, we lose all spatial information. And this information, which cells sit at which place of the tissue and how they communicate, they're very, very important for understanding disease and health states. And if you think about it, a tissue is like a puzzle. So it comprises of cells, which are the pieces, and the genes that are expressed in the cells, they are the different colors. The problem is how to put it all together. So I have been developing computational methods to solve these problems. The first one was to assemble a Drosophila embryo, and we did that by cheating a little bit, by assuming that we know a few images, a small number of gene expression patterns expressed along the fly embryo. If you do that, you can assemble it and you can have a virtual embryo that works really great. And now we have a new method, a new computational method that works exactly on the process that I described before. So we assume that cells which are very close in space share similar transcription profiles. And the method we have tries to mimic exactly this process. This works great for many tissues, colon, liver, embryos, brain, and kidney. Meet me later for details. Thank you very much for your attention.